with cancer, we talk about remission, not cure. And remission is just another word for buying a little extra time. And for the inflammatory diseases like inflammatory lung disease or inflammatory bowel disease, we don't even talk about remission. We talk about trying to improve quality of life. We just try and reduce the symptoms to make your life better, because no one's been able to figure out what causes these diseases or how to kill the cause of these diseases until us. We believe we could cure the incurable.
Almost exactly 2 years ago, Charlie Izzard, a good friend, got in touch with me and said, could I do him a favor? Would I review some scientific papers? And I said, sure, Charlie, what's it about? And he said, well, it's something we're interested in. We'd like your opinion. And the papers arrived, and I read them. And that day, I called him back and said, this is one of the most exciting things I've read in 50 years being in medicine. The technology they have evolved has opened up observations that we didn't know about in medicine, which present opportunities for disease prevention and cure that are unlike anything I've seen in my career. What is shocking about this research to me is that they've now shown that this is a continuum, and the through line for that are these disease-causing stem cells. All of these appear to have this disease-causing stem cell or 2 or 3 different families, and that's true now with these cancers. So it validates what we've thought forever, but it actually tells us why.
The stemECHO technology allows us for the first time that we can clone and grow so many epithelial stem cells from the patient, and for the first time can help us to really understand the complexity of our stem cells in human being.
And you don't have to know anything necessarily about these to clone the stem cells. So I can tell you today, we could start with a new cancer that we haven't really looked at in detail, and in 2 weeks' time, we could have the stem cells for that cancer. You can take that clone and you can grow it forever, which means you can do high throughput drug development.
No one has ever been able to do that before. And the significance of that is that we can fully characterize them genetically, epigenetically, and we've gone ahead and we've been able to understand their role in the pathogenesis of these diseases, the role that they play in causing recurrent and resistant forms of cancer, the role that they play in causing inflammatory airway diseases like COPD, like idiopathic pulmonary fibrosis, like cystic fibrosis, and the list goes on and on and on.
Less than 1% of the cells are causing the cancer to grow and spread. The rest of the cancer cells in the tumor mature and they just sit there. It means that each of these illnesses has a small number, a finite number, 2, 3, 4 perhaps, of disease-causing stem cells, which are the absolute target. Get them and you can cure the patient.
And no one else has been able to do that before because no one's been able to isolate the cells. So the prevailing technology in the field, which is organoids, has a lot of utility. But unfortunately, the organoids cannot isolate individual epithelial stem cells and grow them up in pure colonies of billions of cells like we can with stemECHO. Therefore, you cannot identify drugs that can selectively target them, and you cannot understand the role that these stem cells play in the pathogenesis of these diseases by using the organoid system.
But the decision to move forward is a byproduct of scientific accomplishment, to the point where we now have a number of therapies for different disease types that are ready to go through the IND and phase 1 process.
We're beyond, does it work in A, B, or C? We've actually tested against real biopsies. We've run it through Charles River and other labs. We're ready, and we think the world is going to be ready for this.